Hello, everyone. My name is Miękus. I'm from Nencki Institute of Experimental Biology, Polish Academy of Sciences, and I would like to present my research into corticothalamic relays in mouse models of autism. So, autism is an incredibly broad spectrum of disorders. It is characterized by handicapped ability to engage in social interaction, impaired communication skills, repetitive, pervasive behaviors, and last but not least, disturbed sensory processing. A theory has been proposed trying to unify these symptoms by Markram and Markram called the intense world theory, which draws upon observations that several regions of the brain are hyperactive and hyperplastic in autism. However, no data is available for the thalamus, which may be considered surprising because the thalamus is a central unit processing sensory stimulation, information. And as we know, the sensory processing is disturbed in autism. So in order to investigate that, we used two mouse models of autism, the BTBR strain, which are mice that evince all of the characteristic symptoms of autism, and the FMO1 knockout mice, that has the FMO1 gene silenced, and the inactivation of this gene causes Fragile X syndrome, which is the most common single gene cause of autism. And among numerous connections between the thalamus and the cortex, we focused on a modulatory connection from cortical layer 6 to posterior medial nucleus. This connection serves as a feedback from the cortex to the thalamus, basically telling the thalamus to relay more information to the cortex, so that more information flows to the cortex. In order to investigate this connection, we prepared thalamocortical slices of mouse brains and put an electrode along the fiber in capsula interna, exactly. We then injected a series of 5 electric impulses to these fibers and recorded postsynaptic responses in POM using patch clamp, which is a technique that allows us to clamp individual neurons and measure their electric activity. And this is a sample response we got. This is a chart presenting the flux of current in individual neuron. And we can see that the response to single stimulus, the single impulse, consists of an artifact and the actual response, which in our case was excitatory postsynaptic current, or EPSC. We can also see that the EPSCs form a characteristic pattern getting bigger with consecutive impulses, and this is called frequency-dependent facilitation, something which is characteristic for modulatory connections such as the one we investigate. But when it comes to amplitudes, we saw that they stand out in both autistic strains when compared to their non-autistic counterparts. However, the difference is significant only for the BTBR strain. We then scaled the amplitudes by dividing each EPSC's amplitude by the first one in the series in order to measure the facilitation, so that we can see how many times the response got bigger with consecutive impulses. And still the BTBR strain stands out when compared to its control group. So it turns out that the posterior medial nucleus is overstimulated by the cortex in autism, or at least in the BTBR strain, which means that the gain of thalamocortical transmission is increased. And we hypothesize that, we presume that this means that the transmission of information through POM to the cortex and to the other higher order regions is augmented, which may lead to intensification of tactile sensations perceived by individuals with autism. So I would like to thank you thank you for listening to me, and to Dr. Eva Kublik and Dr. Marek Bekisz, who supervised me on this project.